Right, let's go. Hello guys, welcome to an episode of the Biofiles with my two co-hosts, Dids and Jack, and myself, Ed, and we're going to be taking you through some interesting and topical news stories in the world of biology, which we hope you'll enjoy listening to.
Biofiles!
We might actually have to use that. That was very close to the Mario theme tune.
Yeah, but it was slight spin on it.
But
I don't know what you're talking about. I don't think we're going to get copyrighted for that.
So, without further ado, I'll go first. It's decided. And the topic I found interesting reading this week was something called the Gallery test, which has recently been on the NHS and they're trialling it as a blood sample test to detect for over 50 different types of cancer. And this is quite cool because the blood test, it's called a liquid biopsy, and it looks for circulating tumour DNA. So that's just DNA which has come out of cancerous cells. And they sequence this DNA to try and find mutations which are specific to these different cancers. So a characteristic of a cancer is the DNA, so that's the genetic information. It's not copied properly, and that results in these things called mutations. And particular cancers have particular mutations which you can then, if you can get that DNA, you can sequence it, and that will show you that there probably is a cancer within you. And one of the reasons this is quite big and quite massive, because if you can detect these cancers early stage, it's called stage 1, then you have a lot higher chances of surviving. Whereas at the moment, a lot of cancers which are detected in people, especially in the UK, are usually at stage 2 or beyond, which the survival rates or the successful treatment rates are a lot lower. And yeah, I thought this was just pretty mad, because if you can get a blood sample, which I'd say almost the majority of us have had, for various different reasons, they're pretty easy. I mean, myself, I'm not very good at blood tests at all. I did faint on my first one. I didn't faint on my second one, so we're going up. I think I haven't...
Progression.
Yeah.
Progress, yeah.
I think I have another one coming up soon, so hopefully I won't faint there. And yeah, just something so simple could detect so many different types of cancers, which could have a massive impact on your future treatment and the current rates which treatments are going, and how survival is increasing.
Especially with early detection.
That's the thing.
Yeah.
And that's one of the things which is kind of massive about this, is that if you can detect these cancers early, then you can quickly get it before it metastasizes, which is where it spreads throughout the body and starts not infecting, but amassing in different organs and different places in your body. And if you can stop that, then the chances are huge. But did you guys see that the success rates of a detection aren't as high as you might think?
From your test?
Yeah, from the gallery test, the Grail trial. Yeah.
But I think that's to be expected with any new technology, really, isn't it? So it shouldn't be put to bed just yet. It should definitely be explored, because the idea of catching something early, I really liked your analogy when you said it's like a liquid biopsy. It's so descriptive of what it is. But do you know how much blood is needed? Like, is this like a serial draw from a main vein, or is this like something you can do for an STI test, like when you prick your finger?
I'm not too sure. I can imagine in my head it's a serial draw. So collecting blood as if you were to have surgery and
donate.
Yeah, maybe, well, not to the level of donation, not like pints and pints.
No, you don't need pints. What I mean is you take it in the same manner.
But I guess you wouldn't need too much though, would you? Because if you can, I can imagine, I haven't actually looked into this either, but I can imagine the way that they do it is they amplify the DNA, the mutations. So the way that they go about doing that is with something called polymerase chain reaction, where they know the piece of DNA they're looking for, which is going to be the same in most people because these mutations are so common, and then they can copy that loads of times so that they can detect it, which you think you don't need that much material for that to get loads of it that you can then sequence. So I don't think you'll need that much, but I don't know, a pinprick seems too small, but I'm not...
But you can do a lot with a pinprick though.
You can do a lot with a pinprick. I guess, what concentrations are these in the out in the blood? if they're really, really small concentrations, you expect they would, you might need a bit more just to have you have enough sample to then amplify. But
Yeah, they said half the study having their data stored so they can sequence it later. So they can, cause I think it might have been your one or it might have been Jack's one that I was reading through. Spoiler alert, we're going to go through another one as well. I was looking through and they said 50% of the trial batch will be stored so that if the patients later get cancer, they can sequence it and see if they would have detected it at the initial draw. Is that correct?
that sounds a bit like something I heard on. So I was reading something about the American one, because this is already going out in America, which I think is another thing which is quite big. You can already get this in America. They say that if you have a detection of cancer in your first one, they'll then test you again. They said for free, so I'm not sure how much it's costing people in America to do this. But they made that sound like you then go back and get retested. But I don't know, because if you're storing DNA, that's expensive for them. Two, DNA is like prone to break down, which is why when you get like the in like archaeological digs, when you find some DNA, it's like, oh, we can recreate the woolly mammoth. And it's like, oh, well, you don't really have much DNA there because it's all broken down, but you can try and cross it with some elephant DNA and like fill in the gaps.
like what they did in Jurassic World.
Well,
With Jurassic Park.
I'm not sure how much we're saying that the science in Jurassic Park is legit.
Look, it's true. The CGI didn't exist back in the day to make that kind of visuals.
Yeah, going back to the...
Oh, wait. Sorry. You said how much did it cost, right? The gallery test in America, the list price for the gallery test is $949. Maybe health savings account.
Wow.
A flexible spending account eligible. So I suppose that's some sort of like health finance that America has. I guess in England, they would offer it for at-risk people.
Yeah, so I think the trial at the moment, which is going on, is for people between 55 and 70.
Yeah, sounds about
which
right.
I guess that's where the age of, well, you see these graphs of how likely you are to get cancer, or like how at risk you are, and it just goes kind of like, gets exponential, which is actually quite scary as you go up and up. But yeah, so I'm reading here. I've got written down that the sensitivity for detecting stage 1 cancers is 39%. So for 100 people who have a stage 1 cancer, only 39 people are going to get detected for, which is great that you can detect 39 or that number of the population, and if you can get 39 out of 100 people who have cancer onto treatment like rapido, that's going to be a lot better than no people doing it. But let's segue away. I'm going to go back to the blood or liquid biopsies, which I hadn't really heard of them before, because like a biopsy, whenever you think of a biopsy, it is usually like a bit of your tumour is taken off. So you might have, say, like a dodgy spot on you. They might take a skin graft, and then that is a biopsy of the cancer. That's not a correct definition. I might be wrong there, but it's that sort of thing, taking a sample of the cancer. I think with like lung cancers, the way they do it is they put almost like an endoscope down your throat with a camera on it, and then a pincer, and then they pinch out the cancer and bring it back and do samples on that. Which just sounds horrible.
And by all accounts, biopsies are quite painful.
Exactly, but just imagine it, getting a bit of skin pinched out of you. But if it's a blood sample, where, don't get me wrong, a lot of people are not a fan of needles, but that's like a lot better than taking a bit of, you could argue it's a lot better than taking a bit of your tumour. And that kind of led me down to other ways of detecting like cancers which aren't the standard. Let's take a sample of your tumour and see what's going on. And there's this one called the swallowable sponge, which apparently can detect precancerous throat condition called Barrett's esophagus. Called Barrett's esophagus. It's quite a big capsule, it looks like, and you swallow it, and it has a string on it, and then you like pull it back out of your throat, and then it's a sponge which is like, it then looks like a loofah, like it comes out loads, and all these different spines on the end have your sample on. I don't know if you do that in person, they then like take that out and like test it straight in the lab, or if you then have to send it off somewhere. But just doing this like little sponge which can then detect this cancer or like signs that you might be developing a cancer. I think it sounds pretty cool. And they say this one's quite good because throat cancer is really hard to detect because one of the first symptoms is you can't swallow properly, and that's when it's got too far. Like if it's a tumor big enough where you can't swallow, then that's huge. So if you can detect it early on and like with high risk people, then that's going to be pretty cool. And there's another one I was reading about called I don't know what it's called, but it's like these little cameras, like a capsule camera. So it's like a pill.
Oh, those things are well cool.
You swallow it, and like you kind of all that like nanotechnology thing. You swallow it, and you can go around and try and like look for abnormalities in your body. I don't really understand how it would work though. Like, say it's in your gut, and you have
With
some
the camera,
very good things going on. Like, say you have like big inflammation in your gut. How's it gonna detect for like a camera?
it just records it, doesn't it?
But it's pitch black in there.
I imagine they've got a light on there.
It's just a little search. There's just it is just someone with the remote control.
Yeah, they obviously staff it with small people. And then...
Yeah, true. Put the shrink ray in and all go in. But no, I think these it'll be interesting to see where those technologies are in like 10, 20 years time to see if we are swallowing things to detect. Right, should we move on to the next one? The next topic.
So I was researching, or what I read this week was that Cardiff University may have accidentally cured cancer. They were looking into turning T cells into weapons to fight against bacterial infections, and they, accidentally seem to have found a strain of T cells which are capable of killing pretty much any kind of cancer. But leaving healthy cells completely alone. So it's way better than chemotherapy and stuff that exists now, which is really unpleasant to go through.
That's class. So it's like a really specific treatment just to cancer, whereas chemotherapy knocks out everything.
Yeah, pretty much. Yeah, and they still aren't 100% sure like how it works. So it'll be interesting finding out how these T cells are deciding which cells are cancerous and which ones aren't, and if there's some sort of universal marker of cancerousness that we've been missing all this time that we could use in other detection assays.
I do like the clickbait analogy of the classic tabloid newspaper using all cancers and weapons in your in your analogy. It's incredible, isn't it, that because what the test was looking to kill bacteria, but they were growing bacteria in cancerous cells, and then it was killing the cancerous cells and not the bacteria, or was it killing both?
Well, yeah, it was killing the cancer cells which were infected with the bacteria. So I don't think it was necessarily killing the bacteria themselves. It was just stopping them from taking over cells and being able to live inside them, basically.
Didn't you say that when they removed the bacteria, the treatment continued to kill T cells, the cancers, the tumor cells?
Yeah. That's So yeah, they were infecting the tumor cells with bacteria to see if they could kill them. But then they also realized that the controls they'd included with just regular cancer cells with no bacteria inside them, they were also getting killed. And then they tested them on healthy cells which weren't cancerous, and found that they left them completely alone. So they were like, oh, we may have found something here.
That was going to be my next question. Why were they using tumour cells? Just for ease of culture, or
I think so. Yeah.
That's
They weren't aiming to find a new cure for cancer. Cancer wasn't their like field of interest really. It was just sort of an accidental mistake.
That's
but a happy mistake, I guess.
Which is incredible. I think the fact that it doesn't kill healthy cells, like it hasn't been shown to damage healthy cells, is crazy. Because as we say, chemotherapy, unfortunately, that does kill a massive amount of healthy cells. And you've got the like modern personalized medicine, which is still has like a slight overlap in healthy cells, usually because the things which the personalized medicine are trying to target on the cancer are also found at low levels on a normal cell. So it does have a bit of overlap on normal cells. But this one is, the preliminary research is showing that healthy cells are untouched by this this therapy, which I think for the people who have looked at it and read about it, it's really exciting. like kind of therapeutic industry, or maybe not just that's not a therapeutic industry, because with therapeutics you do get a bit of a cross between they just doing it for the money, but in like the biology community of going forward and curing cancer. And what a few analogies have been saying, it's like a one size fits all, isn't it? Where you could give people like the same drug, like you give people the same vaccine to kill the common cold. If you give people the same drug to kill cancer, then that's going to be phenomenal in terms of like cost and like,
ease of treatment,
the ability to batch produce. Like if you can batch produce something, then that's going to be a lot less.
I mean, even tolerability of treatment, like on the individual, it's so important because, as we mentioned, like previous therapies, like chemotherapy, for example, is a non-specific cancer treatment, which basically just ends up targeting fast replicating cells, and that's why, unfortunately, cancer patients who are undergoing chemotherapy lose their hair, because obviously hair divides rapidly, and so it's targeted by the chemotherapy as well. And a lot of people find that they're just really worn out after chemotherapy because their body just gets obliterated. And I've read reports where they've been trialing new drugs, and it's been efficacious, like it's worked, but the patients are, after asking the patients afterwards, kind of, would you go through it again? They're like, absolutely not. It's just not. To them, it wasn't worth it because it was so intense. So this is fantastic to have that specific treatment, which can be effective and tolerable by the person, because it's only the bad cells that are getting taken care of. It's brilliant.
I think adding on to that as well, one of the sad things about quite a few cancer treatments is if the cancer doesn't kill you, sometimes it can be the cancer treatment which kills you, because it is killing just like so many different healthy things as well, which maybe we won't put that in because it's just a bit doom and gloom, but I think it does need to be known.
It's necessary, yeah, because I mean, I didn't realize for a while that also immune cells were targeted during chemotherapy. So not only do you lose your hair, you'll also lose your ability to defend yourself from like the common cold, because your immune cells get obliterated because they're also fast replicating. And so, I mean, a lot of people, unfortunately, undergoing treatment die from secondary infection. So, is it secondary infection? Is that the way to say it? Basically, an infection that's resulted from a lack of immune system, because the immune suppressants they've been put on so that they don't lose it all leaves them vulnerable. And it's just unfortunate. It's horrible.
Going back to the treatment though, one of the cool things which I was reading about was that they took like different tumours from different people, and the cells were able to attack each of these tumours and like just kill them all. And I think they tested it on like from like breast and lung cancers, which are some of the big ones, skin cancer and bone cancer as well. I think bone cancer is a really horrible drug, a really horrible cancer, which I'm not too sure what the research is and the therapeutics are like at the moment, but I'm quite sure I remember being taught that bone cancer is bad. Whereas like prostate cancer, that has quite a high survival rate comparatively to other cancers. I'm not sure what it's like for bone cancer.
I suppose it's easy. If you look at this, is not scientific, and don't anybody quote me in a conversation or in a paper, but I suppose it's a lot easier to get to like the prostate than it is to get inside your bone, because I mean you've got to drill into the bone, then get inside it, and then deal with the cancer there. That's horrendous. So I mean, obviously
that would still be an issue with this treatment. Say you can treat bone cancer, how are you going to get it to the site of cancer?
I'm not really sure how they actually administer it, to be fair. I think maybe I don't think you would necessarily have to drill into the bone and inject a bunch of T cells or something. I think it would probably just be intravenous or something, and just hopefully it will diffuse into the bones basically and sort everything out.
Did they do any in vivo comparisons in this test?
So, to my knowledge, so far they've only tried it in mice, but they found it was very successful. They found it killed 91 to 99% of cancerous cells.
Wow.
And that's testing in live mice, which
that's
are
huge.
They're fairly similar to humans. Obviously, we won't know if it works in humans until we actually try it, but it's definitely a promising sign.
Did they assess its efficacy on cancer stem cells?
I'm not sure.
Because
That is a big issue, isn't it?
that was what my undergrad dissertation was like looking at. It was like a way of targeting the slow replicating cancer stem cells, which lead to relapse. So when you have these really effective treatments, which are removing 91 to 98% of the tumor mass, there's still 9 or 2% of the tumor cells still in that area. And unfortunately, because cancer stem cells don't replicate fast and they can tolerate a hypoxic environment, which means like there's no oxygen there in the center of the tumor, they can live on and like kind of go undetected during treatment, which is brilliant for the cancer cell, but unfortunately, that will then lead to relapse because the cancer's still there. But where it's at such a small concentration after such a efficacious first initial treatment, it's a real problem because you don't know it's there. You think you've got, you think you've got rid of the lump, so you're all right, and then all of a sudden, a couple of months later, unfortunately, you've got another cancer. That would be really interesting to see if it was effective on cancer stem cells.
Hmm. It kind of like bottlenecks it, doesn't it? It bottlenecks all of the like the horrible, like really super resistant cancers, or the resistant to the therapies, because they're not the ones getting fully killed off. So then when you go into a relapse, and it starts like dividing again, then it's all these horrible like ones which you can't treat, which then come back the second time.
No, but I guess seeing as it's not like chemo or radiotherapy where like it's really a really horrible thing to go through, and really hurts the patient almost as much as the cancer itself. I guess with this therapy, potentially, even if it does come back, you could just give them more rounds of therapy and just keep it under control almost, even if you don't completely eradicate it.
Yeah, I suppose if it's so specific, then yeah, I mean, we don't obviously until they've done this in like a comparable species, we don't know the effect on the individual that this treatment would have. Like, we're assuming that it's fine on everyone because it's only targeting the cancer cells. But yeah, if it's tolerable, then I guess you could. But nobody wants to go. It's like having when you get put on dialysis, isn't it? Like, it's a really effective treatment for kidney failure or impairment, but nobody wants to keep going back to the hospital every 2 weeks to be plugged into a machine. So
No, yeah, that's true. But I guess also people go through like multiple rounds of chemotherapy and
That's true.
radiotherapy. So this would definitely be like a better alternative until we find
100%.
something that can, 100% kill all cancer forever.
Yeah.
But maybe something which we should say is how this kind of goes about detecting cancers and things. Like what they're looking at is MR1, it's called, which is a receptor, and the receptor is something which puts something which is on the inside of your cell onto the outside of your cell. And what they're hypothesizing here is that the cancerous cells are producing different things within inside themselves than a healthy cell would, and MR1 is then presenting these different proteins, they're called, on the outside of the cell, and your body can then recognize this as like, oh, that's not a human protein, that's not a protein which I should be producing, and therefore they can start this antibiotic, or not antibiotic, they can start this immune response against it to try and remove these cells which shouldn't be in your body out of your body. And so I think one of the reasons why it's just attacking cancer cells and not human cells is because it's producing these cancerous proteins and not the human proteins.
It's a really interesting point you brought up about the what would we see in humans, because it often gets overlooked that animal models, or it doesn't get overlooked, but everyone just kind of assumes it as the norm. But animal models are just models; they're not humans, and they have inherent differences. So this receptor which they're targeting on in these models is, like really representative of tumors in mouse. Is it we're using, or rats?
Mice.
Is it
Mice.
Mice, yeah.
In mice? Yeah. And like, will it have the same effect in humans? Will it display the same efficacy? As in, like, as Ed said, is it going to be? Are we going to have any crossover between healthy cells and humans? And like, you just don't know until you use a human model, or I suppose you could use a humanized model. But again, I'm not a massive fan of those. I don't like the idea of those.
Well, maybe,
about an
maybe
organ
this
on a
takes us
chip?
through to our next topic.
Yeah, well, if that's what you want to talk about, guys, then we'll roll on into that. So basically, traditionally cell cultures are 2D, which is two-dimensional cell cultures. As everyone knows, like the human body is three-dimensional. So these 2D models aren't necessarily representative of a 3D environment. So then a 3D cell culture is basically just the same as a 2D, but with the added dimension. The beauty of an organ on chip is you're taking this 3D culture of cells and you're putting it in a model which can now be perfused, which basically means it's flushed with blood or a blood-like substance to replicate what would happen when blood arrives at a tissue in the body and spreads through the tissue, undergoes gas exchanges and nutrient exchanges, and then goes away. You can model that with in an organ on chip. You can also simulate the stretch and flexion response that you get as you're moving around. So obviously your cells aren't static all the time. When you move your hand, open and close it, the cells are stretching. And there's been studies that have found that the stretch and flexion response has an effect on the expression of genes in these cells. So it's really important to include that when you're looking at kind of modeling drug toxicity or therapy efficacy, so how effective a therapy is, because the genetic the expression of genes can affect how effective these treatments are.
What they're doing, isn't it, is that an organ on a chip is a kind of like scaled down version of your organ. So I think for the lung one, a lung on a chip, it's you have a layer where cells are grown, and under that layer you have the layer is a permeable surface like your blood vessels are, and underneath it they're flowing this blood sample through it. But also on either side they've got the vacuums where they can decide how much air is being pumped in and out, which is then representing the air inflow of like when you breathe. And as you said, because it's vacuums, these cause a change in the shape of the cells, which then causes a different genetic expression or just a different change in function.
Influences,
yeah. Which is just huge. So it's kind of just taking one of your organs and scaling it down to we say chip level. Like some of them have been sized next to like a 2p piece. Like they can be very, very small. And one of the beauties of that is because they make them in like see-through containers, you can then look into it. And as you were mentioning earlier, like you can, they're really cool videos. You should definitely look them up if you're at all interested, of a immune cell, like starting off from the blood.
Have you seen it?
Yeah, starting off from the blood, and you can see where the holes are in this permeable membrane, this layer which divides the cells from the blood but allows blood through.
Which is the same in the body, by the way.
Which is the same in the body, yeah. It's all representative.
Yeah.
You can see this immune cell come through. It can then detect this bacteria which is there, which they've obviously put on themselves because they're seeing how the immune response is to bacteria in this sample, and then engulfs this bacteria, and it's able to do that on a chip, which is
and you can live image
happens.
it. You can live image it, and the visuals are fantastic, and they're just such a great way of showing the promise of this technology, and like kind of mapping that response, the mechanistic response, the mechanism of an immune response is so important, because if you can watch it, you can kind of bring up new theories of how things work. The whole modeling the immune system and watching it target, it will infiltrate firstly is brilliant, and then watching it target and locate the pathogens, so the bacteria, is just fascinating.
Yeah, like so that was done has been done like in vitro, hasn't it? Because in vitro is where cells grown in a lab and they then do tests.
It's an in vitro technology. Technically it's ex vivo, because it's taken out of a body. Yeah.
But what I was trying to say is we've like in our undergrad we've seen videos of like a bacteria just going around some cells. Not a bacteria going around. There's kind of a race between a bacteria and an immune cell in like an in
the
vitro.
Classic video. Everybody would have seen it in an introduction to immunology. Must have.
Which I wonder how many people have had.
I'm sure. I think I saw it at school for my first time, like GCSE. So
But that's the sort of thing that we remember as well. I
That's
imagine a lot of
true.
people would be like, oh, what was
Yeah.
that?
It was like a grainy video. It was like black and white.
When there are like 2 pixels in it.
Yeah.
But then like with how Organ on a Chip takes it one step further is it's seeing it not in this thing which is just like a layer of cells. It's
2D,
seeing it in seeing it in a model which is meant to represent what it's looking like in the human, which this sounds amazing, and one for looking at how your immune response, but I think another massive application of it is how like drugs can be you can visualize like drug responses within the body and how a drug goes around and like treats cells or like interacts with cells, and how it's like broken down by your say liver, where a lot of drugs are broken down. Run it through a liver on a chip, run through the drug at a concentration, so an amount which you're likely to see or you inject in the human body, and you can see that this breaks down into this molecule and that's actually really dangerous to the body. Or it might be, actually this then gets broken down into this, which your body can then like treat with, and that's no longer toxic or it's not going to affect the rest of the body. And I guess like one of the massive applications of these organ on chips is it could see a quicker progression of drugs through the phases which a drug needs to go to. So go from being discovered to then actually being put on the market, as they say, where it can be treated with people. And another massive impact of this is hopefully means for animal testing can be eliminated, because at the moment animal testing is seen as necessary because it's the step between testing on just a sheet of cells and then on a clinical trial, because if it kills an animal then it's likely it's going to it's going to affect a human, which isn't nice, but you can see that's needed. That's an ethical issue which we won't go into. And so yeah, if you can do it on an organ on a chip, then that's one, looking at your human cells, so it's going to be more indicative of what's actually happening inside you, and 2, that's animals which don't need to be used in these testings, which is just a win-win, like a huge win-win for everything.
It's in a position where it's working and it's utilised quite a lot. So a lot of the medical or the drug regulators are accepting data from new approach methodologies, which are non-animal methods predominantly. So these are just new technologies which have been produced or kind of thought up, which don't require the use of animals to bring about the same endpoints, so to find out the same information. And the drug agency and the chemicals agencies around the world the majority of them are accepting new approach methodology data to supplement current like data packages for like regulatory approval of their compounds, so be that drug or chemical. This obviously is building up a bank of data for organ on chip regulatory approval as a standalone technology, but we're just not there with the wealth of data at the moment, because you want to submit an application with so much data that no one can say no for the approval. Saying that, it is being used. I was in a conference where a guy from the United States Army's like chemical, biological, radioactive, nuclear warfare research center. I'm not sure the actual acronym, but they use organ on chips to screen for like nerve agents, chemical weapons, the efficacy on human cells or human models, because they appreciate the physiological relevance of the technology, and they're using it because it's the most accurate human model that we can use, which isn't a person. But you can basically connect these chips to make a body on a chip with all these different individual chips. So you can look at lung exposure to a compound, so an aerosolized compound. So basically, it just means in a gaseous form, as if you were to take it in an inhaler or just breathe it in the air. You can then watch that pass through your lung chip, get into the blood system, the blood supply, and then you can track it around the system as it passes to other organs, and then you can look at the toxicity of that compound in those other organs, which is really, really good, because the only other way of doing that is to do it in vivo, which is inside an animal model or a person. Obviously, ethically, you can't do that in a person, and you've seen a lot more ethical consideration with the use of animal models, especially for inhalation testing. It's
You have just talked for about 10 minutes solid there.
Yeah,
Like, just
we'll stop
waffling.
that. Yeah, it's a lot. It's a lot to take
I
in.
hope I can cut out the actual 20 seconds of useful stuff you said in that. And no, I'm messing. I think on that point, are there any other points about anything we've said today which people would like to bring up?
That's good. It's been a long episode, that one.
No, I think we're good as well. Thank you very much for listening to this episode of the Biofiles with Jack, Diz, and myself, Ed, and we look forward to talking next week or next time about 3 more interesting, topical, and I was going to say fun. I don't think fun's quite the right word. Thank you very much. Cheers, guys.